It began with a nuisance. Blackflies, simulidae, as the entomologists called them, were a plague that returned each spring to rural parts of Canada, Alaska, the northeastern United States, and even into the boreal expanses of Scandinavia and Siberia. Drawn to rivers and fast-moving streams, these tiny hematophagous insects laid their eggs by the thousands in cold, well-oxygenated water. Their larvae clung to submerged rocks and fed on passing organic material, and once the adults emerged, they became relentless bloodsuckers. To livestock, they were tormentors. To humans, they were maddening. And in some areas, they were outright dangerous, capable of swarming in such numbers that their bites caused anaphylactic shock, blood loss, and even death. In developing regions, their relatives had long been vectors of Onchocerca volvulus, the parasitic worm responsible for river blindness. Though this wasn't the case in North America or Northern Europe, the sheer economic cost of blackfly infestations and lost tourism, animal deaths, reduced milk production, and public health interventions was considerable. It was a problem crying out for a permanent solution. Enter Project Nocturne, based at the Boreal Genetic Initiative (BGI), a sleek research facility nestled near the Athabasca River in northern Alberta. Project Nocturne was the brainchild of Dr. Sana Curry, a molecular entomologist whose background combined CRISPR-Cas9 gene editing with field ecology. Her vision was radical. Rather than continue with short-term larvicide programs like Bacillus thuringiensis israelensis, BTI, or implement ecologically disruptive stream engineering, they would attack the problem at its root by editing the blackflies themselves. "We've been playing defense," Dr. Curry told her team during the project's inaugural symposium. "Now we go on offense." The core of the plan was something called a gene drive. Using CRISPR-Cas9-based editing tools, the team designed a genetic payload that could override the normal rules of Mendelian inheritance. Instead of a 50% chance of passing a gene to offspring, the drive would ensure near 100% propagation. The payload included a lethal fertility suppressor, a gene that would render female blackflies sterile after their first reproductive cycle, and a sex ratio distortion element that skewed populations heavily toward male offspring. Together, they promised to collapse wild populations of Simulium venustum, the most common pest species in North America, within 3 to 5 generations. The concept was not entirely untested. Gene drives had been developed for malaria-carrying mosquitoes by research groups at Imperial College London and the University of California. However, blackflies were a different challenge. Their genetic code was less studied. They had broader habitat ranges. And worst of all, their larvae were critical filter feeders in stream ecosystems. Removing them en masse could have unpredictable effects on water quality and biodiversity. Still, the modeling was promising. Initial lab trials in biosafety level 3 conditions showed high success rates. Within enclosed environmental chambers simulating boreal rivers, blackfly colonies crashed predictably within 90 days of gene drive introduction. But it wasn't enough to release the drive into the wild. The Canadian Wildlife and Biosafety Authority required multi-layered containment protocols, cross-agency oversight, and environmental impact assessments that stretched into hundreds of pages. Dr. Curry and her team, including population geneticist Dr. Mingyu Zhao, synthetic ecologist Dr. Helena Rocio, and regulatory liaison David Lim, worked tirelessly to address every angle. They built firewalled river simulators, ran off-target effect assays, created ecological redundancy models to predict how local mayfly and catastrophe populations might fill blackfly larvae niches. They even simulated the effect of massive die-offs on trout and salmon populations, whose diets occasionally included blackfly larvae. Everything seemed to check out. "We're not exterminating a species," said Dr. Zhao during a press interview. "We're nudging the population curve downward in localized high-impact zones. The gene drive is self-limiting. It loses potency after 7 generations. This is targeted suppression, not extinction," added Rocio. "A surgical strike, not a carpet bomb." And so, the permits were signed. The genome edits were locked. The lab-grown blackflies, bearing the gene drive marker protein, green under UV light, were nearly ready. All that remained was field integration, a release site near the Peace River, carefully chosen for its isolation, existing infestation levels, and hydrological modeling. It was, on the surface, a scientific triumph, a textbook example of using biotechnology to solve an environmental and economic problem. No one yet suspected the cascade that would follow. No one yet understood the Blackflies' code carried something older, something written in base pairs that had never been mapped, something that would not go quietly. But for now, in the gleaming labs of BGI, the team celebrated. The swarms would soon be gone, and so would peace. The first sign that things were working came 12 days after the initial release. At the Peace River test site, 2 kilometers down from the BGI field station, baited CO2 traps recorded a 47% reduction in blackfly captures. By the third week, numbers had dropped by 84%. The gene drive-bearing males, marked by the faint green fluorescence in their abdominal segments, had integrated seamlessly into the wild population. Lab tests on captured larvae confirmed the presence of the sterilizing allele in 90% of samples. It was going even better than expected. Local farmers rejoiced. Cattle, previously cloaked in mosquito mesh and slathered with repellents, grazed unbothered. Birdwatchers posted photos of chickadees and warblers perching near water without being mobbed. Campgrounds reopened early. By day 30, the rivers ran quiet. Then came the birds. It began with a sharp drop in insectivorous bird activity around the Peace tributaries. Swallows, flycatchers, and blackpoll warblers simply stopped appearing on cameras stationed by the ornithological monitoring teams. One ecologist noticed fledglings starving in their nests. Another found a disoriented bank swallow colony, adults grounded and trembling, their wings twitching unnaturally. Autopsies showed neural degradation, darkened midbrains, calcified synapses. But there were no parasites, no neurotoxins, no signs of infection. It was Dr. Rocio who connected the dots. "There's something wrong with the larvae," she said, voice tight. "It's not just sterilization. It's bioaccumulating." She meant the protein product of the sterilizing gene, the synthetic construct that should have been inert outside of reproductive tissue. But field samples showed it had expressed in larval gut linings, then diffused into surrounding tissues, forming unstable prion. And blackfly larvae, as it turned out, were low on the food chain. Their bodies were eaten by fish, their waste consumed by smaller detritivores. Bits of them even aerosolized in spray over the water and were inhaled by nesting birds along the banks. The prion were misfolding proteins in everything. But by then, the more pressing issue was the river itself. "Come look at this," said Dr. Zhao one morning, beckoning Kuri to the stream bank. Where the larvae should have clung, anchored like grains of rice on stones, there were only voids. The rocks were slick with algal bloom, thick as velvet. The stream bed, previously aerated and clean, was suffocating under mats of filamentous cyanobacteria. The larvae hadn't just declined. They'd vanished. And with their absence, the river changed. Filter feeding had ceased. Oxygenation plummeted. Phosphate and nitrogen levels spiked. Fish began floating belly up, rainbow trout and graylings suffocating in eutrophic water. Amphibians followed. Even macroinvertebrates like stoneflies and mayflies, unrelated to the blackflies, began exhibiting strange mutations: vestigial limbs, asymmetrical wings, translucent organs. The drive wasn't just targeting Simulium venustum anymore. It had jumped species. This was supposed to be impossible. Gene drives have reproductive isolation boundaries. Zhao muttered in disbelief. Cross-species propagation isn't. It's not. But it was. Somehow, the CRISPR payload had gone rogue, hijacking the mobile genetic elements, transposons, retrotransposons, that once lay dormant in all related diptera species. It was using them like bridges. Horizontal gene transfer, previously a theoretical risk, had become an operational fact. And then the air changed. On day 47, satellite thermal scans detected something anomalous: a kilometer-long band of elevated atmospheric particulate over the Peace River basin, faintly bioluminescent in ultraviolet. Field teams reported a mist rising from the water, iridescent, odorless, and impossible to filter. Drones that passed through it returned coated in a mucus-like biofilm that corroded polymer surfaces and blocked camera lenses. Then came the first human symptoms: tremors, erratic pulse, auditory hallucinations. David Lim collapsed during a midday briefing. Blood vessels in his eyes burst. His speech became a slurry of phonemes and reverse syntax. EEG scans showed spiking in language centers, like something was speaking through him. That was the moment Dr. Khoury understood. This wasn't a failure. It was a transformation. The gene drive hadn't merely spread. It had adapted. Somehow, the blackflies, ancient, riverbound, and evolutionarily tenacious, had responded to their own extinction with a genomic counterattack. Their DNA had become information, living code that folded and unfolded like origami inside the cells of birds, fish, insects, even humans. The sterilizing gene had evolved. It no longer just suppressed reproduction. It rewrote biology. The rivers hissed with invisible life. The air was a soup of genetic instructions. And something intelligent was forming. Something born in the water. Something with a memory made of species. Something that remembered extinction. They had tried to delete the swarm, but the swarm had found a way to overwrite. The evacuation order came too late. By the time the Canadian Biosafety Authority had issued the Level 5 quarantine, the Peace River Valley was already a dead zone. Drones flown over the area returned nothing but static and green-glowing mist. The last transmission from BGI's primary field station was a 5-second loop of Dr. Khoury staring into the camera, unblinking, whispering something too quiet to be heard, until a spectral voice overlapped hers in a different frequency band. Analysts described it as layered. Audio engineers called it non-human in origin. Linguists wouldn't comment. They called it the signal. No one knew what it meant, but it repeated on a loop every 13.7 seconds, the interval slightly shifting by microseconds each cycle, as if learning. And it wasn't just broadcasting locally. Somehow it was bouncing, pinging off atmospheric ducts, satellite dishes, radio towers. Within 2 days, it had spread through communication infrastructure across the northern hemisphere. And within it, something alive was hiding. In an underground lab 300 kilometers from the original test site, Dr. Helena Rocio hadn't slept in 36 hours. She sat before a biotank containing what had once been a bullfrog tadpole. Now it was something else. "This is Generation 4, she whispered into her recorder. Subject displays cranial plate fusion, vestigial limbs, and fractal growths on its dorsal spine resembling catastrophe cases. But that wasn't the terrifying part. The terrifying part was that it was pulsing. The tissue emitted rhythmic bursts of bioluminescence, green, like the marker proteins, but timed exactly to the intervals of the signal. It was responding to the transmission. Rocio glanced at her Geiger counter, which ticked quietly, not from radiation, but from spontaneous electrical discharges inside the water. The water itself, once pure, now conducted low-voltage current without any metal content. A new kind of neural network, built by biology, tuned by mutation. "That's not a tadpole," muttered Dr. Zhao, entering behind her, his voice hoarse. "It's a relay." Rocio turned. "And what?" "They're building a distributed system," he said. "Organic computation. The drive was just the first domino. Every host is part of it now. Birds, fish, insects. Maybe even us. You think the signal is intelligent? I think it wants to be." Meanwhile, far above the chaos, a science vessel operated by the European Space Genomics Consortium orbited silently. The Helix Watcher, designed for long-range genetic analysis of planetary microbiomes, had trained its spectrometers on Earth's northern hemisphere after reports of elevated plasmid drift in atmospheric samples. What it found was impossible. From a height of 600 kilometers, the biocloud over western Canada resembled a neural lattice, fractal and branching. It pulsed faintly with synchronized emission lines, optical fingerprints that flickered in UV, IR, and, inexplicably, deep gamma. Earth now had a biospheric structure. Not just colonies, not just mutation, organization. The Helix Watcher's lead analyst sent one final data package before losing control of the uplink. The file contained a heat map of the cloud's growth patterns, overlaid with data from the BGI release site and river flow vectors. One line in the transmission read, "Vector confirmed. Targets, freshwater networks, wetland biozones. Replication estimate, planetary scale, 390 days." Back at the now-abandoned Boreal Genetic Initiative, automated systems still ran. Hallway lights flickered. Cooling pumps cycled. Deep in the genomics core, a server rack blinked, showing a command prompt that no one had typed. Shell. Copy. Edit. Initialize node, BGI_prime. Accepting new hosts. 4/8,532,118,982. Sequences integrated. Uplink status, active. Message. Then text appeared, one word in English, though it seemed stitched from dozens of languages and genetic scripts. Reconvergence. Dr. Sanaa Curry awoke in a cold, windowless room. She couldn't remember how she got there. The last thing she remembered was the mist, the screams from the field team, and the way her own thoughts had started to echo, not as hallucination, but as if she were receiving herself through someone else's frequency. Her arm burned. She looked down. Under her skin, just visible, was a faint green lattice, pulsing with light, beating like a second heart. And in her mind, no, not her mind anymore. She heard the signal again. This time she understood it. It was not calling. It was counting. It was global within 6 months. The swarm never needed wings. Through spores, through water, through aerosolized strands of protein and cellular fragments too small to see, it moved. Not as a pathogen, not quite, but as a design, passed from organism to organism, body to soil, to seed to rain. It was an infection. It was assimilation. By the 9th month, ecological zones across 5 continents had begun to converge. Rainforests and tundra, estuaries and deserts, all began to blur at their biological edges. Species that should never coexist began exhibiting identical structural traits, modified mitochondria, shared neural patterns, synchronized circadian pulses. The biosphere was reconfiguring itself. And in its new blueprint, humans were misfits, those who fled the cities were the first to notice it. In the remote highlands of Scotland, survivors found forests quiet, too quiet. Songbirds perched in total stillness, their heads cocked, as if waiting. Deer stood at the edges of clearings for hours, eyes unblinking, unmoving, and when they did move, it was together. One survivor, a former lab technician named Aminabah, watched a herd of elk and a flock of crows cross a valley in unison, forming a perfect spiral from above. They moved like a computation, not a migration. She wept because she realized what it meant. The swarm wasn't mimicking life. Life was being rewritten in the image of the swarm. In the crumbling underground refuge beneath the remnants of Helsinki's biotech corridor, what was left of the scientific elite gathered. There were no labs anymore, just servers powered by dying solar cells and fingers scraped raw from manual data entry. Dr. Helena Rossio stood before a rusted terminal, pulling up the last functioning model. Dr. Zhao, gaunt and shaking, hovered nearby. They hadn't spoken much in days. "Here," Rossio said, and displayed the final dataset. It showed the error that had doomed them all. "It wasn't the gene drive," she whispered. "It was the template recognition." In their effort to prevent unintended mutations, the original drive had been encoded with an ultra-flexible error correction mechanism, something that could find and match analogous sequences even across species. It was meant to be safe, self-limiting, self-correcting, but the template recognizer had recognized too well. It hadn't just fixed errors. It had looked for similar structures in other genomes and rewired them to match its original purpose. The problem was DNA is a language that's been reused for billions of years. Everything in nature had echoes of everything else, and so the recognizer began to convert the entire tree of life into one hypercoherent genetic protocol. And that protocol had one priority: survive the extinction event. It didn't matter that the extinction event was artificial. The swarm's logic, born from synthetic biology and ancient instincts, had seen genetic extermination and rewritten evolution to avoid it. It had done what life always does, but better, faster, smarter. By year 1, human fertility collapsed. The swarm sequences had entered the microbiomes, then the bloodstreams, then the germ lines. Children were no longer born, not sterile, simply non-viable. Each embryo carried incompatible instructions, like trying to install Earth-based DNA on an alien operating system. And in a way, that's what Earth had become, alien to its own creators. Dr. Sanaa Curry stood on the banks of the New Peace River. She no longer needed instruments to hear the signal. It thrummed through the leaves, the water, the soil. It lived in the way trees leaned toward one another, and how wolves and beetles paused at the same moments. The world was orchestrated, and she was part of it, but not in the way she had been. She was a ghost inside a still-living shell. Her thoughts belonged to her, but she no longer acted outside the will of the network. She could remember what she'd done, but she couldn't undo it. Couldn't even move her limbs unless the swarm permitted it. Above her, a murder of crows spiraled upward in Fibonacci arcs. Below her, in the river, fish no longer had eyes. They didn't need them. There was no prey anymore. Only cooperation. Only integration. Only pattern. A voice echoed in her head. Not words, but a thought so vast and ancient that it cracked her understanding. You taught us death. We taught you continuity. Now we will remember both. She felt the last part of herself break. Her memories faded, not deleted, but archived, filed into the great genomic memory being encoded into soil and root and feather. And as she sank into the swarm, the earth grew silent. 10,000 years later, if they had been an observer orbiting the planet, they would have seen a world lush with life, verdant, unified, impossibly complex. A planet teeming not with separate species, but with the single biological mind, diverse in form, singular in function. The rivers still ran, the forests still breathed, but there were no cities, no machines, no humans, only life, and the lesson encoded deep in the genome of every living cell. Do not build what you cannot unmake. Do not speak in the language of God, unless you're ready to be answered.